Hello there, how are you guys? Welcome to my presentation on invisible influence in biocomputing. My name is Sukhjeet Singh, and today I will explore the fascinating world of biocomputing, focusing on its background, advancements, and global impacts. We will delve into the potential of biological computing, the challenges it poses, and its implication for the future of science and technology. Let's begin with a brief overview of what biocomputing is and how it's revolutionizing the field of biology. We'll move to the next slide now. As the name suggests, biological computing is designed from living cells inside of electric wiring and signaling that uses chemical inputs derived from protein and DNA. Just like modern day computers, biological computers can process and respond to various data and statistics in a similar manner as computers performed in 1920. While biological computing in the 21st century has a long way to go before they provide accurate results, as today personal desktop, the fact that scientists have been able to complete a logic gate is a commendable achievement in science today. You won't say that once the biological computers are programmed with a single biological cell or molecule, it is extremely easy to replicate billions of cells and molecules only at the cost of lab tech time and nutrient solution. Moreover, researchers also believe that biocomputers in the future will be more reliable than their electronic counterparts to help bodies survive even when millions of cells are dead inside. On the contrary, uh, Guizhou states that although biocomputing provides seamless opportunities in biological research, the executed programs in the machine can create an unpredictable biological environment that can impact the timing, nutrition, and conditions of the cell. In computational biology, gene finding refers to the process of predicting areas of genomic DNA that encapsulate genes. Genes can be protein coding and RNA in nature, however, methods for detection may vary in protein coding and RNA genes. As per AVSCC, finding genes has significant significance in biocomputing and genetics as it helps unearth large infected sequences which may result in spread of disease. Similarly, gene prediction necessarily identifies core elements of the genome such as splicing sites, intron, exon, and regulatory sites for gene encoding and decoding. If these fundamental elements are not identified correctly, the functional gene may blind with the abnormal proteins, further resulting in a contagious sequence. Therefore, gene finding is also necessary to distinguish to find a difference between coding and non-coding regions of the gene. In addition, Mostavi state that gene finding is considered as one of the most significant breakthrough in the history of genetics and biology because it, in, it individually identifies genes and link their function with it so that biocomputing can be applied in different fields like metabolomics, proteomics, genomic study, and functional genomics. All these functions can be used in detecting genetics mutation, treatment, prevention, and diagnosis. Hence, gene prediction can be of great advantage for researchers, biologists, and scientists. The second subtopic is protein sequencing. As stated by Shen, protein sequencing is necessary for medicine and biology as it allows the analysis of amino acid to confirm the exact structure of protein in the body. When amino acids are not sequenced or appropriately identified, the identification of new proteins can be restricted, which further result in wrong phylogenetic reconstructions. Furthermore, protein sequencing can also be applied in various biological processes such as disease detection, understanding ancestral history, and identification of protein drug market. Initially, when biocomputing was not introduced in genetics, protein sequencing was a critical challenge for researchers. Also, protein sequencing was primarily used to obtain genome information for medicinal use. However, after the evolution, protein sequencing was not only used for medicinal purpose, rather the process became popular to produce generic variants that may lead to disease or risk of disease development in future. Therefore, it can be concluded that protein sequencing is an important in biocomputing to assess genomic information. Multi-skill modeling and single-cell sequencing has been one of the most recent scientific advancements in biocomputing. According to Yen, before biological computing was introduced in genetics, scientists used single-structure modeling methods to understand complex structure. Due to this reason, the assembly of over 200,000 atoms and dynamics could not be constructed properly. However, multi-skill modeling has been a major breakthrough in biocomputing as it allows millions of atoms to combine together and generate massive genetic data that not only has statistical significance but also help scientists understand complex structure of bacteria, viruses, and other cellular bodies. Along with this single-cell RNA sequencing, another significant achievement in genetics and biocomputing. It allowed researchers to manipulate individual cells instead of extracting them from a larger population. Besides this, handling enormous amounts of data such as RNA sequences without efficient tool resulted in strong sequencing and decoding. Therefore, single-cell sequencing was given priority in biocomputing to extract and sequence RNA individually. The same can also be linked with modern society where biological big data can be individually stored in .pdb format, which is easily readable by both computers and humans. Moving to the next slide, we will talk about simulation and network verification. Identifying networks has been one of the greatest benefits of biocomputing globally, as it benefits biological researchers in conserving energy through parallel computation. Besides this, biocomputing is a highly efficient and relatively inexpensive process where not only large quantities of samples can be produced at the same time, but specimens can be cultured without any additional operations. As stated by Michels, the invention of biological computers is regarded as the invention of 3D mini brains that entirely mimic the structure of a brain. Hence, the same characteristics can benefit the majority of research centers globally by enhancing the computational capacity of computers and the human brain. Reconstruction of computational phylogenetics. Until the 20th century, reconstruction of phylogeny was critical to detect abnormal sequencing and genome variation due to lack of computational abilities. However, with technological advancement and innovation in genetics, phylogenetic reconstruction became easy, which is now considered a global benefit for analyzing genetic sequence and molecular data to reconstruct the evolutionary history of diverse organisms. On the other hand, phylogenetic reconstruction is also an advantage for establishing a suitable relationship between relative and common ancestry. Biocomputing has excellent potential in understanding protein nucleic acid recognition, investigation macromolecular complexes, and improving multi-resolution structure prediction, and using interactive modeling. However, the biocomputing instrument is accompanied by global challenges. that impact scalability and reliability and pose major challenges in genome comparison and, and identification of polymorphic genetic variation. Genome comparison. Genome selection and comparison involve three major parameters: quality, quantity, and diversity, through which biospecimens can be tested in the lab. However, gathering diverse genome specimens through biocomputing is a crucial challenge in genetics and genomics research. As a result, a huge portion of the global population can miss out on the transformative benefits of genomic comparison needed for effective therapies and early diagnosis of disease. Polymorphic genetic variations. The use of single nucleotide polymorphism is a standard process in biocomputing that is designed to immediately respond to variation in drug treatment. Nevertheless, identifying a single polymorph is difficult in biocomputing due to genetic basis diverse and uncertain nature. Moreover, when polymorphic genetic variation are not processed and evaluated appropriately, the variant or gene may decode an abnormal expression, generating the protein sequence in DNA. Therefore, polymorphic genetic variation can be global challenge in biocomputing. Moving to the next slide. It's a reference page, and thank you for listening to my presentation. We hope you liked it. So that's all from me now. I will take a leave now. Thank you. Bye bye.